Good day everyone, my name is Jefferson Isapasa from BSN1 Abdella. And for this session, we are going to be dissecting a pig's kidney and will locate its major parts, as well as we are going to discuss the blood formation and the formation of urine. Let's get started.
Let us first examine the external part of the kidney. The kidney is covered in this yellow-white fibrous layer called the perirenal fat capsule. It also known as the renal adipose capsule, is a layer of fatty tissue that surrounds the kidneys. It serves as protective cushion for the kidneys, helping to isolate and support them within the abdominal cavity. On this side of the kidney, we will see the structures where blood and urine enter and exit the kidney. This part is called the hilum, and they are divided into three parts. First, the renal artery. This transports blood coming from the aorta to the kidneys. It branches off from the abdominal aorta and enters the kidney at the renal hilum. Second, the renal vein, which transports the oxygenated blood to the inferior vena cava. Third is the ureter, which is a hollow tube that connects the kidney to the urinary bladder. This ureter can also be identified by this large amount of fat tissue attached to the kidney. Now, we carefully split the kidney in half lengthwise, from the side to examine the interior structure. Let us now identify the three main regions of the kidney and the structures that are present within them. First, the renal cortex. This is the outermost part, and it contains the glomeruli and the renal tubule, which are the site for blood filtration. It contains most of the functional units called nephrons, which are responsible for filtering the blood and producing urine. As you can see, it is shaped like a letter C, and it surrounds the other regions. Second, the renal medulla houses the nephron loop, commonly known as the loop of Henle, and is where absorption occurs. We can also see the renal pyramids, which collect and carry the produced urine from the nephron's renal tubules. There are also several renal pyramids visible, which are divided by renal columns. We can also see medium-sized veins in the renal columns that transport blood filtered by the nephrons to the renal cortex. The white component is known as the calyces, and it is divided into two separate parts. First is the minor calyx, which collects the urine from the renal pyramids. Therefore, it surrounds the renal papillae of each pyramid. The second is the major calyx, where the urine passes through before going through the renal pelvis. Next region is the renal pelvis. This collects and stores the urine. This is the funnel-like structure that will later on help the urine go through the ureters. Now that we know the three main regions and parts of the kidney, let us dive deeper into the blood flow through different renal vessels and the three major processes of urine formation. Blood filtration and flow begins when the renal artery found in the hilum region that separates into segmental arteries. This will also branch out to the interlobal arteries, which will pass to the renal column, going to the base of the renal pyramids, specifically the arcuate veins. Then, it will separate again through the cortical radiate arteries, which goes to the renal cortex. Then, it will enter the glomerulus, which will start the filtration of the blood. Once the blood is already filtered through the nephrons, it will exit to the efferent arteriole and to the peritubular capillaries, going to the cortical radiate veins, and will go through the renal pyramids again. Starting at the base, or what is called the arcuate veins, which connects the interlobal veins, and lastly, it will exit to the renal veins that is connected to the inferior vena cava. Glomerular filtration. This is the starting point of urine formation, which takes place in the renal corpuscles, which are composed of the glomeruli and the Bowman's capsule. The filtration process starts with the blood coming from the afferent arteriole and enters the glomerulus. The filtrate coming from the glomerulus is now collected in the Bowman's capsule, which is then called glomerular filtrate. This filtrate is propelled to the renal tubules, where reabsorption, secretion, and concentration processes are further refined into urine. The second stage is tubular reabsorption. This process involves four structures of the nephron in the renal tubules. First, the proximal convoluted tube, or simply proximal tube. This conserves the essential components that have been filtered by the glomerulus. Almost of the glucose is reabsorbed in the PCT. Water is reabsorbed through the tight junctions between the PCT cells. This movement is stimulated by the osmotic gradient that was created by the active transport of solutes like glucose and ions. Second, the loop of Henle. This makes the filtrate more concentrated through its descending limb by absorption of water. Consequently, the ascending limb is permeable to water. However, it reabsorbs sodium and chloride ions. Third, the distal convoluted tubule, DCT, or simply distal tube. This reabsorbs the electrolytes under the control of aldosterone hormone. Fourth and last, the collecting ducts. The ADH increases the collecting duct's permeability to water, which allows the reabsorption of water and makes the urine more concentrated. The third and final step is the tubular secretion, which involves the DCT and the collecting ducts. This stage is important as it gets rid of substances such as excess ions, drugs, toxins, and waste from the blood that were not filtered enough in the first process, which is glomerular filtration.